Hello, everybody. Thank you for joining this webinar by the International Academy for Clinical Hematology. It's a great pleasure and an honor for me to be with you here today, where we will be discussing some important issues related to the therapy of relapsed multiple myeloma, where I'll try my best to give you an update about the latest research evidence in this field. These are my disclosures relevant to this talk. The learning objectives of this webinar are listed here. I'd like first to highlight the principles of therapy for relapse refractory multiple myeloma, including the objectives of the therapy, when to start the therapy, and which factors to be considered in the decision-making process. Then, in the second part, I'll go through the different available options, highlighting efficacy, but also management of side effects. These options will include the discussion of second-generation proteasome inhibitors, monoclonal antibodies, but also the more novel, other innovative options, including CAR T cells. For the sake of introduction, I would like to highlight here that we have seen over the last few years a clear change in the paradigm for the management of relapse refractory myeloma. Historically, at time of first relapse, because of the very few available options, it was thought that the game is over. Now we have so many options that we can see patients more and more at different relapse phases. And actually, at each relapse phase, a new agent can be, or combination can be tested, and this will allow for a certain duration of response or remission. And at the end of the day, when we combine all of these relapse phases and remissions, we have now more and more patients surviving for many years, beyond 10 or 15 years. Actually, beside autologous stem cell transplantation, we owe the major advances in the field of multiple myeloma to some important family of drugs. One major family are the proteasome inhibitors, and you can see here the mechanism and the structure of the proteasome, of the immunoproteasome in the cell. And I think everybody's familiar with bortezomib, which was the first proteasome inhibitor to be approved. But now we will see the results of the so-called second generation proteasome inhibitors, such as carfilzomib, but also ixazomib, which is the first oral proteasome inhibitors. There are also other proteasome inhibitors being under development, like marizomib. I would call them the third generation proteasome inhibitors. The other family of drugs which really made a significant advance in the natural history of multiple myeloma are the IMiDs. And I think everybody is aware of thalidomide, but now we have the second generation lenalidomide, but also pomalidomide. And more IMiDs are currently being developed. And these are quite amazing anti-myeloma agents because they have a very complex and not fully known mechanism of action, acting directly on the myeloma tumor cells, but also acting on the microenvironment and we know very well the influence of the microenvironment on the growth of plasma cells. IMiDs have also an immunomodulatory effect, acting on some key immune effectors, and probably this is the mechanism of action in the maintenance setting when using IMiDs. From a clinical standpoint, what are the treatment goals when we see a relapsed multiple myeloma patient? I think first, and this is why patients come to see their physician, you need to reduce the active symptoms burden, but you have also to aim to achieve the deepest disease response possible, namely complete remission, but more and more, I think we should aim to achieve MRD negativity, negative MRD. And it's not only about biological MRD, but also it can be about imaging and negative PET scan, for instance. At the end of the day, we would like to prolong patient progression-free, but also overall survival. However, we should not forget that during all these steps, we should prevent any additional morbidity and preserve the quality of life of the patient. And this is becoming more and more a matter of concern because we have new agents that are rapidly being introduced into the market, and sometimes the experience is not very strong, and the practicing physician need to describe all the side effects that are being observed by the patient. When should relapse treatment be started? This is a very difficult question, and there is no universal answer yet. We usually claim that treatment of relapse, especially biochemical relapse, like when you start seeing the monoclonal component increasing progressively, we should not act too early, but also not too late. And it's a true dilemma, and there is no clear answer for this. However, more and more, I think the community is moving towards sort of an early intervention, because I think we should try to avoid organ dysfunction. Obviously, it can be quite difficult because organ dysfunction can be kidney failure, but it can be also bone pain. And these symptoms are not the same. And we have to understand and describe the pattern and clinical features of patient relapse. Next question is about which factors to be considered when deciding on a relapse therapy. We have two groups of factors: the patient-related factors, such as age, and this is important because you can treat a patient at age 60 and the patient will relapse at age 66 or 67, and in the meantime, performance status will not be the same. Some comorbidities, some events may have occurred during these years. Bone marrow reserve is an important issue to consider, especially if patients received a stem cell transplantation, 1 or 2 transplant, or even if they have received long-term therapy with IMiDs, or if they have received alkylating agents in general. Renal function, and we know this is the weakness of all our myeloma patients, but also pre-existing toxicity. If patient has developed a peripheral neuropathy to thalidomide or has thromboembolic events, then obviously that will limit your options when it comes to other agents that can generate peripheral neuropathy. And of course, quality of life considerations, but also patient preferences. I think more and more the patients are being very active in the decision process and we should give time and listen to their wishes and to their hopes. Treatment and disease-related factors. Cytogenetics, we know at time of diagnosis, is really important, but we also now know that at time of relapse, that can be also critical. The components of the initial therapy. Obviously, it's not the same story if you are resistant to a given therapy versus you are still sensitive. Duration of initial therapy, but also efficacy of initial therapy. Obviously, a patient who has been in remission for 3 or 4 years after a given therapy, this is a totally different feature compared to a patient who relapsed like 6 months after the end of therapy. On one hand, you may have an indolent group. On the other hand, you can have a more aggressive group. Relapse under therapy is something crucial because it highlights the emergence of some resistant clones. So let me start first by reviewing the role of proteasome inhibitors in multiple myeloma. Of course, I will not go back to all the bortezomib studies. We'll move now to the second generation proteasome inhibitors. And the second generation proteasome inhibitor that was tested in this ASPIRE trial is the carfilzomib. This has been published a few years ago in the New England Journal of Medicine, updated during the last ASH. This is a randomized phase 3 trial comparing carfilzomib, lenalidomide, dexamethasone versus standard of care lenalidomide, dexamethasone. So a triplet combination, 28-day cycles, given for a maximum of 18 cycles versus a continuous therapy with a standard of care len-dex. And the primary endpoint was progression-free survival, and you can appreciate that progression-free survival was significantly higher in the KRD, in the carfilzomib arm, versus the len-dex arm, with a hazard ratio of 0.55, which highlight a 45% improvement, 26 months versus 16 months in the latest update. Another important feature, I think, is about overall survival, and this trial could show almost 8 months of median improvement in terms of overall survival in favor of the triplet combination with carfilzomib, lenalidomide, dexamethasone versus lenalidomide, dexamethasone. In terms of side effects, there is a safety signal when using carfilzomib in relation with cardiovascular events, whether it comes to dyspnea, hypertension. This is an IV infusion, carfilzomib, and you usually hydrate the patient. Cardiac failure, ischemic heart disease, pulmonary embolism. Taken alone, the incidence is relatively low, but higher than the len-dex alone. So patient selection, especially when it comes to cardiovascular risk factors, is something of great importance when using this triplet combination. The other proteasome inhibitor, second generation, is ixazomib. And ixazomib is the first and only approved oral proteasome inhibitor. And that was approved thanks to this so-called TOURMALINE MM1 study comparing the triplet ixazomib, lenalidomide, dexamethasone to lenalidomide, dexamethasone. Again, the same standard of care. One important feature is this is a double-blinded study, placebo-controlled. So I think the design is rather very nice, and the treatment was given until progression. The primary endpoint of the trial, like in the previous trials, was PFS, progression-free survival, and you can appreciate on this red curve that the ixazomib len-dex combination was significantly superior to the lenalidomide dexamethasone combination. When it comes to side effects, non-hematologic adverse reaction, one needs to pay attention to some gastrointestinal side effects, especially when it comes to diarrhea or constipation, nausea, vomiting. But these are usually side effects that are easily manageable with symptomatic therapy, and the oral approach can be very convenient in many old or frail or fragile patients. Looking into the proteasome inhibitors-based combinations, we would like to highlight this recently reported results of a phase 3 randomized trial, the Optimism trial. Actually, this is a very interesting trial which combined the first generation proteasome inhibitor, bortezomib, to pomalidomide. We know pomalidomide is a highly active IMiD. So this is the PVD combination versus the VD combination. VD is another standard of care approved, although it can be less popular than Lendex in some countries. And the primary endpoint of this trial was progression-free survival, and you can appreciate that the PVD triplet combination proved to be superior to the VD combination, 11.2 versus 7.1. And this is a rather short follow-up, but I think these results are very promising, especially when looking into patients who received it frontline. And this triplet combination is coming now to enrich the therapeutic armamentarium. And you can see on this slide the PFS in patients who received one prior line of therapy at 20.7 months versus 11.6. This is similar to what we have seen with some of the previous trials I have mentioned. In terms of side effects, obviously you are using an IMiD, so cytopenia can be there, neutropenia, although this will not necessarily translate into a significant side effects, but we should not neglect in these patients the incidence of infections, especially pneumonia. Again, this is another major weakness of multiple myeloma patients in general. And I think one of the suspected drugs when it comes to infections is usually dexamethasone, actually. So we should not forget that many of the side effects in our patients are usually related to the high dose dexamethasone, but also to the cumulative dose of dexamethasone. We are using bortezomib, even if it is used subcute. There is a certain number of patients who will experience some peripheral neuropathy, and this should not be neglected because it can significantly alter their quality of life. That was the proteasome inhibitors based new combinations, at least 3 triplet combinations available and approved with some positive and interesting results. For the sake of time, I will not discuss the doublet combinations with proteasome inhibitors, and there are very attractive data when using the combination of carfilzomib dexamethasone versus bortezomib dexamethasone. This is the Endeavor randomized phase 3 trial, which showed superiority of the carfilzomib dexamethasone combination. Here I would like to spend some more time on monoclonal antibodies, because we know this is a major family of drug that is clearly making a difference in the natural history of multiple myeloma, not only in the relapse setting, but also soon, I think, frontline. Just before going into the details of the currently available monoclonal antibodies, I would like to highlight to this audience that monoclonal antibodies were tested in multiple myeloma for more than 15 years, actually with little success. And you can see on this slide a non-complete list, I would say, of different monoclonal antibodies which were tested, including the famous rituximab we all use in lymphoma, with little success, either because of lack of efficacy or because of the side effects. And it was until 3 or 4 years ago when we have seen the results of the first active monoclonal antibodies in myeloma, and we have now 2 agents available and used worldwide. First agent is elotuzumab, which was approved based on this ELOQUENT-2 study. So similar to the previous studies I have discussed, elotuzumab was combined with lenalidomide dexamethasone and compared versus lenalidomide dexamethasone standard of care. Elotuzumab is a monoclonal antibody that has no single agent activity. It targets CS1 on plasma cells, which we also call now SLAMF7. And when you look to the extended follow-up, the extended 4 years follow-up, looking into the primary endpoint of the trial, PFS, there is a significant advantage when using elotuzumab Lendex versus Lendex. There's also a survival advantage in these patients. Obviously, these results, these figures are rather modest compared to what you would expect in with some other drugs. Another important aspect about elotuzumab is about its safety. It's a very safe drug, but it acts very slowly. So the profile of patients who can benefit from this combination needs to be carefully selected. Then we have the anti-CD38 monoclonal antibodies, and there are already several candidates, either chimeric or fully human. Daratumumab was the first to be approved. Isatuximab is being currently under investigation in phase 3 trials, and hopefully at some point it will be approved. We have also the MOR202, which is less advanced in terms of development, but hopefully at some point will be available. So I will mainly focus on the results of daratumumab because this is the most widely used and available monoclonal antibody, but also approved, not only in the relapse setting, but also in the frontline elderly setting, as it was done recently by FDA and EMA for the Dara VMP combination. So daratumumab has a very interesting mechanism of action. It targets the CD38 antigen, which is highly expressed on myeloma plasma cells. It has a direct anti-tumor effect, killing the plasma cells. It has also an immunomodulatory effect because we know CD38 is expressed on different immune effectors, but also it has an immune-mediated effect. And to make a long story short, it is likely that beside the direct killing of the plasma cells, the use of daratumumab is allowing to create a sort of an microenvironment, sort of an immunostimulatory microenvironment, allowing to get rid of the regulatory and suppressive cells. And that is definitely something of great value in cancer in general and in multiple myeloma. Another important feature about daratumumab, it's the only novel agent beside dexamethasone or melphalan, which has a single agent activity. That was the first approval, and these were the first trials of the drug. You can see in highly advanced and refractory patients, you can still achieve in responders as a monotherapy a very nice median PFS. Interestingly, even in patients who are in stable disease, the results are better than in those in progressive disease, and this is maybe can it be explained by the immunomodulatory effect. The major daratumumab trials and combinations are the so-called POLLUX and CASTOR trial. You can see here the design of the POLLUX trial. This is daratumumab combined with Lendex versus Lendex, again, standard of care in the relapse setting. You can see the key eligibility criteria. The primary endpoint, as usual, was progression-free survival. And in the updated results of this trial, which were actually published a couple of weeks ago in the journal Hematology, median follow-up is around 33 months. You can appreciate there is a dramatic improvement in favor of the Dara Revdex arm versus the Revdex. And you can notice that the Revdex arm is rather a strong arm because you have a median PFS of 17.5 months, which is really one of the highest ranges you can achieve. The hazard ratio is 0.44, something around 65% of improvement for 56% of improvement, in these patients. Another important feature about this daratumumab Lendex combination is the very high response rate, more than 90%, but also, and this is quite a unique feature, the capacity of this combination in the relapse setting to achieve in some patient MRD negativity. So usually one would like to see MRD negativity in the frontline setting, and here you can see it even in the relapse setting. So definitely this is good news because we do have a body of evidence suggesting that MRD negativity is a surrogate marker for survival. And actually, we do not have the final results, the updated results about the overall survival. Nevertheless, we can consider PFS2 as a surrogate marker for overall survival. PFS2 is when patients relapse after a given treatment and you try to salvage them with another treatment, and you can see the results of PFS2 are also in favor of the Dara Lendex combination. When it comes to adverse event, these are mainly in relation with infusion-related reaction. This is why pre-medications are needed. One should also pay attention to cough. Most of the patients will have some cough, especially in the beginning of the infusions. When it comes to other side effects, these are more in line with the side effects we see when we use lenalidomide dexamethasone. The other registration trial for daratumumab is a combination of daratumumab with bortezomib and dexamethasone. So combining it with a proteasome inhibitor, Dara Veldex versus Veldex. Again, these are standard of cares, same design, progression-free survival as the primary endpoint. It's important to note that the control arm was given for a limited duration, whereas the experimental arm, Dara Veldex, was given for continuously. You can see the results with a median follow-up of 27 months, a little bit more than 2 years. There is a clear advantage in favor of Dara Veldex, whether you consider the ITT population or patients who were treated in first line. Of course, one need to be a bit critical about these results because obviously this bortezomib dexamethasone as a control arm is not the ideal arm and not necessarily the most widely used. Nevertheless, it's clear that these results are appealing and can be useful in many patients. Similar to the POLLUX trial, we do have a PFS2 advantage for Dara-Valdex compared to Valdex, and this is definitely a surrogate marker for an improved overall survival with this combination. And I think this is important because if you have improvement in overall survival, that can suggest that even at time of relapse after these therapies, you can salvage these patients with the novel agents that are becoming available. But the story of treatment possibilities will not stop with the drugs I mentioned, because there are already new players coming into the field. Selinexor triplet-based combinations are being tested, and venetoclax triplets are being tested, and hopefully will become available in the near future. So the field is moving really very rapidly, and this is clearly very exciting. But it's not only about drugs, about pharmacologic agents. Now we have true immune and cellular therapies for multiple myeloma. And here I'd like to spend a few minutes discussing the first results of the CAR T cells in myeloma. Just as a summary, for those who are not very familiar with the CAR T cell technology, the idea is to redirect the TCR towards the tumor antigen for any given tumor. That would allow you to overcome the HLA restriction, which we know is a major barrier, and at some point you would like to induce a significant number of high affinity T cells that can target and kill the tumor cells. However, one needs to bear in mind, especially when it comes to autologous CAR T cell therapy, that this is a multi-step procedure. It's not like having a vial or a pill that you give to a patient off the shelf. You have first to identify the specific tumor antigen. So this is going to be disease specific. You need to be able to produce a high affinity antibody. You need to genotype this antibody, and you need to be able to transduce your lymphocyte, whether they are autologous or allogenic. Obviously, in the development process, you would like first to assess the efficacy in vitro and in vivo. This is why mice models are extremely important. And one important feature beside efficacy, it's about safety, because of course, you would like to avoid cross-reactions against the standard panel of normal tissues. So it's not as easy as it appears when it comes to selecting the potential antigen in a given tumor. So in myeloma, currently what is being discussed is the CD138 antigen, also called Sandican-1, kappa light chain, CD19, also this is unfrequently expressed on myeloma cells, but the first proof of concept, first case report published in the New England Journal of Medicine was CAR T cell directed against CD19. SLAMF7, which is targeted by elotuzumab, I mentioned a few minutes ago with the ELEGANT trial. Actually, that can be an attractive target for CAR T cells. And the most widely tested now is BCMA, the B-cell maturation antigen. So why BCMA is so popular, and why the myeloma community is focusing on this antigen? It is expressed by plasma cells and some mature B cells. It is universally expressed. So in terms of efficacy, you would expect that you will be able to target all the tumor cells. But interestingly, when you look into the mice which are knockouts for BCMA, they are healthy with normal B cells, but reduced plasma cells. So in other words, you would also expect maybe a good safety profile, because the cross reactions are not very important. So these are the different CAR T cells under development in multiple myeloma, and you can see the different major companies. involved in this development. It's not only about culturing these cells. This is a very complex process, but also it needs to be performed in certain environment to ensure the quality of the manufacturing of the products. For the next few minutes, I think I will focus only on the most mature results that we have seen in the literature and in the different meetings. These are the results that were obtained with this CAR T-cell construct, the BB2121. It's a phase 1 trial. You can see the design of this trial, including 2 phases, a dose escalation phase and a dose expansion. Patients are screened. Of course, you need to do an apheresis, leukapheresis, to collect the lymphocyte. You manufacture these cells. If they are of good quality, they are released. Patients are going to receive some lymphodepletion. This is crucial because obviously if you would like your infused CAR T cells to survive and proliferate, you need to create the right immunological environment with fludarabine and cyclophosphamide, and then you infuse your CAR T cells, and of course patients are followed. To be simple, I think what we already know based on the results of this very important phase 1 trial is that patients can achieve, they are highly refractory patients, more than 7 lines of therapy, so you can guess how advanced is the disease. With this, you can achieve almost 1 year of PFS, which is very impressive in a fully refractory population. And it's not only about PFS, it's also about MRD negativity in a group of patients, 16 patients, where here you can have up to 18 months of PFS. Definitely a relapse continue to occur, which means that we're not completely eradicating the myeloma cells. But again, it has to be evaluated compared to the nature of the patient, refractory patients who were included. When it comes to side effects, I think with CAR T cells in general, one needs to pay attention to the cytokine release syndrome, which is a very severe syndrome in some cases and can kill the patient. And it has some very well-established clinical features. So training on follow-up of these patients and getting experience is crucial. Neurotoxicity is also another important feature. And unfortunately, we do not yet know what is the exact pathophysiology and what kind of intervention you need, in contrast to the cytokine release syndrome, where we know that the anti-IL-6 receptor tocilizumab can allow to revert the situation and allow the remission of the CRS. In this phase 1 trial, it proved that the incidence of CRS and neurotoxicity, at least the severe forms, were rather very low compared to what has been seen in other trials with acute lymphoblastic leukemia, for instance, or lymphoma. Now there is an ongoing trial, the so-called KARMA trial, including more patients using these CAR T cells. So that was the, I would say, the summary of the different available options. Obviously, for the sake of time, I wasn't able to go into all details of everything, so I do apologize in advance, but I'm more than happy to discuss this at a later stage during the discussion and by email. So how are we going to proceed in the clinical practice? I think all of us would agree today that treatment of relapsed refractory multiple myeloma patients is very challenging because we have a lot of new combinations. They are emerging. They are moving rapidly to the first line of therapy. I mentioned that daratumumab is a key drug in the relapse setting, However, we know that it is approved now in the frontline setting. So how are we going? What is going to be our behavior in those patients who were treated frontline with daratumumab at time of relapse? Of course, we need the phase 3 randomized trials to get the approval, but these trials take a lot of time, and by the time they are finalized and available, the landscape is quickly changing. And one example is about the use of lenalidomide dexamethasone as a control arm. When the trials were designed, actually, we didn't have the approval of lenalidomide, and very few people expected to use lenalidomide continuously for maintenance. We have to deal with the type of relapse. I think not all relapses from a clinical perspective are the same. You have the aggressive relapse, medullary compression, kidney failure, hyperglycemia, and you have less aggressive, more indolent, like anemia, bone pain. Early versus late relapse, the story is not the same, and this has to be taken into account. Number and type of prior lines of therapy, especially when it comes to sensitivity versus refractoriness. Assuming you have a patient who is receiving frontline lenalidomide maintenance and this patient has relapsed under lenalidomide, well, obviously, one needs to take into account the refractoriness to lenalidomide. Cumulative toxicities can be limiting in these patients, especially, I mentioned cytopenia, for example. Patients who were received double transplant, received a lot of IMiDs, then that will limit your options. Comorbidities, this is a moving target because multiple myeloma patients, the median age at diagnosis is 70. So this is the age where you accumulate comorbidities, and comorbidities will increase with age, but also with the different treatment you receive, because by themselves they can generate comorbidities. Patient expectations, I think there are totally different expectations from one place to another, from one patient to another, but also the patient expectation can change during the course of the disease. When they retire, I think their expectation having the disease after retirement is not the same during your active professional life. So we should not also forget the access to novel drugs and sequences, because obviously this is not a medical parameter, but we have to live with the issue of access, because all drugs are not available in all countries. Even in the most developed and rich countries, you may not be able to get access to all the drugs. So you have to design your sequence based on availabilities and pharmacoeconomic parameters. This is why I didn't allude to this, but I would like to highlight in my conclusion the issue of retreatment with the same drugs, especially if you have patients who were responders to a given line of therapy and remained in remission for several years. I think these patients can benefit from a retreatment process, whereas when it comes to early relapse or aggressive relapse, probably switching to a new class of drugs and new combinations is going to be of great importance. With this, I'd like to thank you all for your attention wherever you are. I hope you have enjoyed this seminar, this webinar. We will be able to take a few questions, and I already started receiving a few questions. I can see them on my screen. So I will try to answer. I will read the question, and then I'll try to answer. For the sake of time, we won't be able to answer all questions, but definitely there can be a follow-up by email. So first question I can read: what is the role of second transplant in treating relapsed multiple myeloma? This is an important question. I didn't show results, but definitely salvage auto transplant if you have an available graft, especially if you have a patient who is responding to a salvage regimen. But also, most importantly, I believe these patients should be sensitive to melphalan, to an alkylating agent. And by saying sensitive, I think 2 years is a minimum requirement for response to a first auto, then definitely we can consider the use of a second auto in a fit patient. I have another question here. Is there still a role for chemotherapy for relapse refractory multiple myeloma in the era of novel therapy? My short answer is yes, there's always a role for chemotherapy in highly refractory patients, in extramedullary disease, in plasma cell leukemia. And of course, if you don't have availability or access to the novel drugs. So short answer is yes, one needs to keep in mind that the old-fashioned classical chemotherapy can be of benefit in some patients. I have a third question here. Why old, not specific acting dexamethasone is so important in relapsed myeloma and this novel combination? This is a wonderful question, actually, because everything we do in myeloma, we combine it with dexamethasone, whereas we know very well that this is a key player in the toxicity. Unfortunately, many of the myeloma agents do not work alone as single agent. I mentioned, for example, elotuzumab, but actually, if you look to lenalidomide results without dexamethasone, they are less good compared to with dexamethasone. The same story is true for bortezomib. So I think we're not ready yet to design dexamethasone-free regimens, although this should be our goal. We already started, thanks to the ECOG trial, by using 20 milligram of dexamethasone instead of 40 milligram. And my personal advice to everybody is do not hesitate to use lower doses of dexamethasone, 10 milligram or even 5 or 4 milligram, especially in elderly and frail patients. There are other questions which are coming into my screen. Do you expect a renal toxicity failure in case may targeted CAR T-cell therapy, and how would this potential toxicity be dealt with? I think this is quite difficult to answer, at least from the trials using CAR T-cells with directed against BCMA. I'm not aware that this is a feature, that this is a toxicity that appear to be a major one. However, we should not forget that the patients who are receiving now the CAR T-cells are the most advanced patients, and usually in these advanced patients, kidney function is rarely normal. So there are a few other questions keep on coming. I think I propose to all of you to have a follow-up by email. So if you send your questions to the IACH address, definitely we will be able to handle them. With this, again, I would like to thank you all for your attention. I hope you have enjoyed this webinar, and I hope you will become loyal to the monthly IACH seminars. And I can already announce to all of you that the next seminar is going to be scheduled on the 14th of November by Dr. Miguel Perales from New York, who will address the role of CAR T-cells in lymphoid malignancies. Thank you very much, and wherever you are, have a good day, good night, or a good start. Thank you very much. Goodbye.